My name is Jeremy Jaime, and today I'm going to be talking about the different types of viruses, replication of viruses, and difficulties in eliminating viral diseases. So, the first type of viral disease that you have is double-stranded DNA virus, and examples of this virus can be adenovirus, papillomavirus, polyomavirus, herpes virus, and pox virus. For the next type of viral disease, you have single-stranded DNA. Single-stranded DNA, an example of this could be parovirus. And then you have double-stranded RNA, which an example could be reovirus. So, so one thing to take note of is that all RNA viruses have an envelope, and that viruses are not considered or classified as eukaryotes or prokaryotes. So for the replication of viruses, there's two different ones. You have lytic and you have lysogenic viral replication. So for the first one, lytic, the first step for this is going to be the attachment of the virus to the cell via the tail fibers. And so for the second step, you're going to have the entry of phage DNA and degradation of the host DNA, in which the next step would be synthesis of the viral genomes and proteins. And this would cause the virus to be self-assembled to the capsid protein. And so the last step would be in which the lysis bacterium releases the virus or the capsid bursts, and now you have multiple viruses free roaming, going to the next cell to infect it. So for the lysogenic cycle, it's a little bit lengthier than the lytic cycle, but there are some similarities to it. So you have the same thing, the tail fiber attaches to the cell, the virus injects capsid content or its DNA genetic material to the cell. But when this happens, the cell fails to recognize that the virus is a foreign substance. So it actually accepts the virus's DNA into the chromosomes. And so when this happens, mitosis of the cell happens, and this would cause the cell to divide and pass on that virus's genetic material or the genome to the new cell. And after this happens, the cell enters the lytic cycle.